All right, so I decided to start from the beginning because people asked me, what my role was in discovering dominant negative interactions. And actually, the history of dominant negative interactions goes back to the 1920s. Uh, in Drosophila, actually Colombia, the Drosophila geneticists there found these mutants that they called negative trans um, trans interfering mutations. So there would be a mutation that would interfere with the function of the wild type. But then and then they kind of sat around, and then in the 1960s, Benno Mullerhill, working on the lac repressor, actually stumbled on it when he was trying to isolate mutations that are a lot more familiar with you, that were promoter mutations that overproduce the lac repressor. And these were these mutants he tried to isolate. Their lack IQ, and the reason for isolating them was to try to purify the repressor. And in the course of doing that, he stumbled on these trans-dominant negative mutations. Sort of towards the end of my graduate career, I got interested in, I actually stumbled on this because I was learning how to make deletion mutations in the transposon I was working on, which was TN5. And the point of the study was to try to find out what the minimal sequences that were necessary for transposition. And in the course of this, I stumbled on this phenomenon where it turned out that in trans, derivatives of TN5 were actually able to inhibit newly introduced TN5 transposons from integrating into the chromosome. And at the same time I was doing that, work was also going on in Bill Resnikoff's lab by Reed Johnson. And so then the two of us, our two labs then published these back-to-back papers in Cell that describe this phenomenon in which there's naturally occurring dominant negative interference that actually is made by nature to regulate transposition. This is the first, I'm pretty sure this is the first evidence for naturally occurring dominant negative interference. There aren't, I think still in the literature, there aren't that many examples of it. So, but this is just kind of an interesting thing. And as a result, you can see what you can see that we used a question mark because I was actually so insecure about this. We certainly had evidence from the literature that you could isolate mutants that behaved in a trans-dominant fashion, but actually this being a naturally occurring phenomenon seemed was a cause for insecurity. And so Ben Lewin, who was the publisher at Cell, seemed to love papers in which people ask question marks. You don't see it anymore, obviously. So anyway, so this was the system. So the system they developed over a number of years was to analyze TN5 transposition off of phage lambda. And the lambda that I used was unable to integrate into the chromosome and also had multiple deletions in it that allowed lambda to package the transposon. And there was just a simple system, and then we just analyze for transposition onto the chromosome. And so then, as I was telling you, I was making deletions, and I had found that all you needed for transposition was one of these repeats. So the way TN5, the structure of it is as two inverted repeats. The one on the right I'll concentrate on because that made two intact proteins that had two different start codons separated by about 50 amino acids. And you could see on gels, now I don't think I'm going to show you a gel, you could actually see these two proteins being made. The left repeat had some mutations, so it wasn't making intact versions of these proteins. And in between was kanamycin resistance, that's the neogene. And so what I found by making deletions is that only this right repeat was necessary for transposition, and it had these two proteins here. And so I was messing around with this, and I stumbled on this fact that when you do an infection with a lambda and this plasma is in trans, it actually interfered with transposition onto the chromosome. And so again, this is the way this looks. There's these two proteins, and fortuitously, there was a restriction site right here. Remember, we had limited molecular biology techniques then, and so what I was able to do actually was to construct something where I inserted a oligonucleotide that had an XBA1 site in it. That XBA1 site actually introduced a stop codon in frame with the larger protein, and so I was able to make strains that just had this single protein 2 there, and that also was totally fortuitous. And so this shows you the phenomenon. So we could get transposition occurring at about almost 1 in 10 to the third infected cells here if we just had just a cloning vector in trans without the plasmid there. But if you had both protein 1 and 2, which is kind of an interesting phenomenon, so they were overproducing these 2 proteins, or if you had only protein 2 alone, by 2 to 3 orders of magnitude, you were reducing the transposition frequency. And then what we ended up showing actually here was by using LAC fusions, these were protein fusions. Again, this was still fairly new technology at the time. And using these various derivatives of that actually, it didn't matter whether the protein was produced in trans or there's no protein produced in trans. There was no effect on the actual translation or transcription of the protein. So then we came up with this model, and it was based on this, and this is actually from my discussion that I wrote here. This has been shown that some knockout mutations in the LAC repressor have a dominant phenotype. In these cases, if mutant subunits of LAC repressor mix with wild type subunits, the resulting tetramer is inactive. And so then the idea here was what's coming off of the transposon were a mixture of the full length protein and the truncated one. And if the full length protein multimerizes, you're able to get an active transposase. And this is actually what people use when they're doing the Nextera TN5 transposition, and they're doing that in vitro. It's using this pure full-length protein, obviously, without the shorter one there. And then the plasmid then is making either the interfering one, if we have this protein 2 here, or a mixture of the two. And then that's allowing these mixed multimers, and then that was preventing transposition. Okay, so dominant negative. The term dominant negative, if you look in 1982, that's when this paper was produced. We didn't use the term dominant negative, but that there were absolutely no papers published at that time in 1982 that used the term dominant negative. And then you can see this huge increase as people were using this as a tool. And so this is the reference that I had. This is the Muller-Hill and Gilbert. And it's kind of lost in here because what people are more interested in is that it overproduces LAC repressor. But dominant negativity, that use of dominant negative mutants became popular about 5 years later when Ira Herskowitz then published a paper. It was a review article in Nature in which he encouraged people to use dominant negative mutations as a way of analyzing particularly mammalian cell function. Because remember, in 1987, there wasn't really a way to easily knock out genes in mammalian cells. But if you overproduced a protein that had inhibitory activity, and again, Herskowitz posits that the best ones to use are ones which make mixed multimers, that would be an effective way to actually analyze mammalian gene expression. So that's 83, 84, 85. So that's about right here. And then you can see after he wrote this paper, there's this huge increase in papers using that term dominant negative mutations. So at any rate, so that was kind of one of the things from my graduate thesis that people were curious about, sort of where I kind of fell in the whole landscape of discovery of dominant negative mutations. And I think I got it pretty well correct here. All right, so then this is just before I left Stanford for Tufts. So I went to Stanford to do my postdoc in the Falkow lab. And when I entered his lab, people probably read this little article I wrote. He says, "Why don't you clone Invasin into E. coli?" And I naively went along and did that. And so that was based on this observation. And so it might even seem a little bit weird to you. The people who work on Yersinia think of it as an anti-phagocytic organism that prevents phagocytosis. But the way I got into this, and the reason why Stanley believed very strongly in the idea of uptake into mammalian cells is this work that Dan Portnoy did in his lab. And here's an EM of this, in which he takes bacteria, he actually grew them at 28 degrees, which I'll show you is kind of key, and then infected them into HEP2 cells. HEP2 cells are probably contaminated with HeLa cells. And then did an EM, and what you can see is there are all these bacteria inside the cells. And so Stan said, well, why don't you clone this? Because nobody at the time knew how bacteria get into mammalian cells. And of course, this is really what I wanted to work on. And so even though I didn't tell him that's what I wanted to work on, it's what he suggested I did. So I was I remember leaving his office shortly after starting my postdoc, feeling really happy because he told me to do exactly what I kind of wanted to do anyways. All right, so again, this is the kind of the weird thing about this is we think of Yersinia as being anti-phagocytic. So remember, it has this type 3 secretion system that translocates the YOPs, and the YOPs interfere with phagocytosis. And that's really true at 37 degrees. However, if you grow at 28 degrees, you actually don't see that. And this became clear in this. This actually is I think this is one of the first papers I published after I got to Tufts. And actually, I'll actually show you where I did that work. So if you look at this, bacteria are growing at 28 degrees. This is fully virulent organisms. And then you just pop them onto a monolayer. You can actually see bacteria inside mammalian cells. This is with Yersinia pseudotuberculosis. And Portnoy done it with Enterocolytica, but we see exactly the same thing. Then if you grow them at 37 degrees, then the anti-phagocytic YOPs then are translocating into the host cell, and that's interfering with uptake. So you see this big dichotomy. And then the question then is, what's going on in vivo? And it's clear actually that there's a couple pathways going on. I'll talk to you about this in a few minutes. But one pathway that people have believed in was that when bacteria are ingested, they enter into regional lymph nodes after Yersinia is ingested, and then they enter through M cells. And then, so then the question is, what's Yersinia actually doing? Because we know that the bacteria grow within mesenteric lymph nodes. And so we kind of use this idea that maybe there was this bifurcation, so that, early on after infection, the bacteria haven't grown much in the intestine. They could enter into regional lymph nodes. And then once they start growing in the lymph node, the anti-phagocytic YOPs turn on, and then that prevents neutrophils from phagocytizing them. And I'll show you the evidence for that in a couple of minutes. Okay, so based on this then, I just did this experiment that Stanley and the other people went crazy for, but at the time I just thought it was just kind of, just a stupid cloning experiment, which is, I just took Yersinia DNA, partially digested with Sal3A, ran a cesium chloride. I remember these goopy DNA cesium chloride gradients, and then this was then I used a plasmid vector, which people don't use anymore. So what these plasmids are is they have a packaging site, or a COS site, for phage lambda. And then that allows you to package very large pieces of DNA. And of course, what we thought was that the ability to enter into animal cells would be really complicated. And so we assumed, actually both Stanley and I assumed that would mean that we require getting a lot of genes into E. coli and getting them all expressed. We envisioned a very complicated process. And then once they were all expressed in E. coli, then we could get the E. coli entering in the mammalian cells. And so to maximize that, I did this partial Sal3A digestion that gave you fragments between 40 and 60,000 in nucleotides long. And then ran them out on a gradient, and then ligated them into this plasmid vector, and then used an in vitro packaging extract that I made from phage lambda, and then packaged them infected in E. coli. Now this is, it's amazing how inefficiently this worked. I mean, I got about 500 colonies coming up in this packaging, but each one of these, each one of these things that were packaged had about 1% of the Yersinia chromosome. So I probably got a pretty good representation of the chromosome with just about 700 colonies. But I guess it might have been lucky just the same. So I wasn't that comfortable with so few colonies, but I still pooled them. I introduced them onto monolayers. And then everybody's familiar with the gentamicin killing assay, but I didn't do that, okay? And so what I did is I was worried about it. So what I did is I just washed off all the bacteria from the monolayers, and then I just released the bacteria associated with the animal cells by adding Triton, because E. coli is resistant to Triton. And then what you can see is that I got the surprising results. So this is actually a map of the cosmid. And, it's a fairly small plasmid. It was about 45,000 base pairs. And then the only thing I knew how to do was did saturating TN5 mutagenesis on this plasmid, and then mapped where all the insertions were. And I was very surprised to find out that all the insertions were actually in a single gene, and that we could clone this fragment, and that was sufficient to allow bacteria to enter into mammalian cells. Well, Stanley thought this was wonderful, and I didn't totally understand why. And he said, well, nobody would have thought of using mammalian cells as a selection to select for something with bacteria. And I guess that was true, because it hadn't been done before, but if you're a bacterial geneticist, it's just another way of doing a selection. All right, so then it came to Tufts with that clone, basically, and not much else. And this is my lab, and this is my first technician, Debbie Voorhis. I recruited her from Mount Holyoke College because she worked with a former graduate student from Tufts who had recommended her. And then this is John Leong, of course, and this is Rob Fournier, who I hired off of my second grant. I was lucky to get 2 grants fairly quickly after I got to Tufts. And so then we fairly quickly published this paper. We identified Invasin with Debbie on it, right here. And then John came in the lab, and then he made a bunch of proteins. And then this was actually the first paper from the lab that was totally due to work that only came from the lab. And this was the demonstration that actually Invasin is a cell binding protein. And so the way I did these experiments, and I forgot about this, but this was actually really cute. So the way the experiment was done is, one of the ways we did this experiment was, I ran protein on a gel without boiling the protein in SDS, and then simply blotted them to a filter. And you can see Invasin is a really schmutzy protein. This is the wild type protein, and this is a deletion mutant that is missing the carboxy terminal of, and John had shown that the carboxy terminal was required at the time I did this. And so the carboxy terminal was required, so this mutant was unable to bind mammalian cells. So this is just an immunoblot. No, maybe I got this wrong. This must be, yeah, no, this must be, I got this wrong, sorry. This is definitely a cell binding blot right here. So this must be a deletion at the immunoterminal end of the protein. So at any rate, so this is then a cell binding blot. And what I did here is I just transferred to nitrocellulose, actually it's Mobilon filters. And then in the absence of SDS, and then just took the filter and then probed the filter with HEP2 cells. And then after allowing that to probe with the HEP2 cells for a couple hours, just fixed the cells, and then stained them, then stained the cells. And then what you can see here is what happens is this isn't in color, but the blot is this fairly pale blue color. And then the cells actually stain dark blue. And I think it was, I can't remember which dye I used to stain the cells. With some protein dye. It was not Kamasi blue. But anyway, and if you looked under the microscope, actually, you could see these individual cells actually spreading on top of the protein. And so then John did this experiment because he, at the time he was in the lab, he was isolating monoclonal antibodies, and he found some that blocked the ability of bacteria to enter into mammalian cells. And what he showed here is that this protein, which blocked binding, was also able to block entry of the bacteria. So this is introduction of, these are controls right here, the bacteria entering into mammalian cells. And then here's antibody that's directed against the carboxy terminal of Invasin, and that's blocking the entry of bacteria. And the way we do this experiment, this is a gentamicin killing experiment, where we infect bacteria for an hour or two, we wash off, and then we add gentamicin for a half hour or so, which will kill extracellular bacteria, but the ones inside will survive. And then we played out colonies on a petri dish. All right, and so then this eventually led to the crystal structure, and this is just want to point out that Shelley Brown was a technician in my lab. Actually, she was the first technician I recruited from Oberlin. And she purified the protein. We had a really nice collaboration with Juji Hamburger and Pamela Borkman's lab. All four of us had quite a close relationship with each other. So it was really a fun PhD project that she did in Pamela's lab. And so one of the things she did was crystallize the carboxy terminal of Invasin. You can see right here what it is. It's a series of immunoglobulin-like domains. And then at the head of this is a C-type lectin-like domain. And this piece right here, this 192 amino acid fragment, is sufficient to bind mammalian cells. And right here, what Juji's pointed out here is D911. This is the critical aspartate, which John Leong had shown during his postdoc, was the critical aspartate necessary to bind mammalian cells. And then another postdoc, Laura Saltman, had identified these other two residues. And what Juji's pointing out is this looks very similar to fibronectin, which binds the receptors, the same receptors as Invasin does, as well as the critical aspartate, which is also found in fibronectin, which binds the critical receptors. And those receptors are integrin receptors. And I'll get to you how we showed that, okay? Yeah, so this is just the cell binding domain here. All right, so so John's name's on this paper because he gave me protein. So this is I think this is the last really series of experiments I did in the lab before I basically retired to my office. And so John actually gave me the constructs to do these experiments. So what John showed was that the carboxy terminal was able to bind Invasin, and fortuitously, I can't remember why this was, we added a couple amino acids to the end, and that totally blocked the ability of Invasin to bind mammalian cells. And so what I did here is and Paula, this will be interesting for you here, I covalently linked these proteins to Sephiroth beads, and then incubated them with mammalian cells. And what you can see is these beads that have the isoleucine leucine added on don't bind mammalian cells. These do, and they spread out. And this is actually what it looked like on those filters when I probed the filters with mammalian cells. So that looked good. So I thought right from that we could probably use this assay to identify the receptor. So the strategy I took at the time was I was reading the Pierce catalog, and they'd come out with these reagents that were biotinylation reagents that were impermeant to mammalian cells, and they'd had them out for about 3 or 4 years. So I thought I'd use them. And so what I did was to take mammalian cells, and I grew up like, buttloads of this stuff. So I take like 10 T175 flasks. And they make, and I put them all together, and they make up a cell pellet that weighed about 1 gram or 1.5 grams. And then would label them, wash them off, then label them with a biotinylation reagent that was impermeant. So it would biotinylate lysine residues. And then I made a dodecyl sulfate, it's octoglucoside, extract. So that's a gentle detergent that retains functions of proteins. And then bound the entire extract to one of these beads, okay? And this is an example of the flow through. So these are all the biotinylated proteins that came through. And so right here, what I'm doing is I'm probing with streptavidin, and that's alkaline phosphatase links, so that we could, detect what was going on here. And then what I did here was I eluted with EDTA because I found out that the divalent cations were necessary for cell adhesion. So I figured if I added EDTA, I would be able to elute the protein off of the column. And what you can see here is if you elute off the column that has Invasin carboxytermalon, we get this very distinct pair of bands coming off. And if you use this derivative over here that has the isoleucine and leucine on there, there's nothing coming off. So these were all very beautiful blots, as you can see. And these are all different cell lines. And actually, each one of these cell lines had a different, so when you probe these with they were the same size as integrin receptors. So immediately I thought they were integrins. And I probed with antibody against different subunits, different chains of integrin receptors, and was quickly to find that they all had the beta 1 subunit, and they all had different alpha subunits. And these all different but related integrin subunits, integrin receptors that were eluted off. And this is also a fun experiment. I went over, so it turns out that back in the old days when you didn't have to file IRBs, you could just, call up the Red Cross and get some outdated platelets to be delivered to the blood center at Tufts. And so I just call them up, and the next day, like, these platelets would show up. And so what I did is I just walked across the street to the medical center, picked up some platelets, then made an extract. There's a huge amount of platelets, right? So here I'm not biotinylating or anything like that. This is just a Comasi blue stain gel. And so I basically did the same experiment as before. These are platelets. And this RGD elution is interesting because RGD binds the same region of integrin receptors as Invasin does. It turns out that Invasin binds so tightly that this peptide's unable to elute Invasin, but EDTA does. And so I basically did the same experiment I did previously, except instead of this being a blot, a streptavidin blot, what this is a Comasi blue stained blot. And then Mike Byrne was there even then. And so I cut out what I thought was the alpha subunit of the integrin receptor, and then he sequenced the amino terminal end, and we found out that this was actually the fourth receptor that we'd identified. Okay. And then finally, this is another experiment I really liked. I was able to reconstitute the receptor into phospholipid vesicles and get this to work in vitro. I used, the way I did this is I actually used labeled phospholipid in this. So I used a small amount of phospholipid that was doped. And so then this is actually radioactive counts, but I knew how much I was able to, because I knew how much lipid was in there, I was able to get a good determination of the amount of radioactive counts per nanogram of lipid. So, so this is a fairly clean experiment. In this case, RGD elution actually was able to work on these phospholipid vesicles. And the other thing I showed is that the phospholipid vesicles actually bound better to Invasin than they did to native substrate fibronectin. You can see the scale is different. Okay, so then this scale right here stops right over down over there, and you require a lot more. So clearly Invasin was actually a much better receptor for the integrin receptor than fibronectin was. I think this is the last time this experiment was ever done in my lab. Okay, so now let's get back to, was it true actually that the bacteria enter into M cells in the intestine? And this is the evidence for it. So, so this other group who were really good cytologists showed that M cells have integrin receptors on their surface, and you can't see that in the lumen of the intestine with other cells. So what these guys did is, this is a Peyer's patch, and what they did is they probed with a lectin that recognizes fucose, which recognizes M cells. and then probe with antibody against beta-1 integrin. They can see that actually where the M cells are is where you see beta-1 integrin. And then when there's no M cell there, there's beta-1 integrin in these cells, but they're not on the luminal end. They're actually on the basal end. So basically the idea is that bacteria cannot bind anywhere in the intestine via invasin except where the M cell is. And then, about the same time, I guess this paper was actually published after Andrea's paper, but it's a much nicer picture than what we were able to get. And this is some of the experiments that Andrea Mera did. And so this actually shows a Peyer's patch, and you can see that the bacteria actually enter into M cells. This was shown by a number of other groups as well. And you can't see entry anywhere else along the intestine. It's just like, it's just totally clean. And so this kind of explains what's going on here where we're getting entry. Again, now the question is this what's really going on? And this is when, like, some like bad data came out. I wouldn't say bad data, like bad results came out in the literature that, that sort of questioned the idea that Invasin was even involved in anything involved in pathogenesis. And so these were like dark days in the lab. And so one of the things that actually, solved this issue was actually looking at this particular model, which turns out to be, I'd say, 80% wrong. And so the model that everybody had was that bacteria start systemic disease by entering into M cells, and then they go into the lymphoid Peyer's patches and then grow in the Peyer's patches and then disseminate into other tissue sites like the mesenteric lymph nodes and the liver and spleen. And everybody believed that. I certainly believe that. And people still to this day go around spouting this, in spite of the fact that we have established that this can be a minor pathway or a non-existent pathway. And it's been verified actually in multiple pathogens now as well. So when people say like the normal pathway for how intestinal pathogens cause systemic disease is to first colonize in regional lymph nodes and then go deep into tissue sites, you really got to question that, because actually the Peyer's patches actually turn out to be a really good place to interfere with progress of the bug spreading to other tissues. Okay, so now what was the negative and what was the positive? So what was the negative data? So the first negative data that came out was that in Yersinia pseudotuberculosis, Wolfwatz and colleagues showed that if you knock out Invasin and do an oral infection, the LD50 is the same as wild type. Okay, so that was bad. So people started laughing at me, you might imagine. However, shortly after that, within 5 years, Virginia Miller's lab came up with a, kind of a result that people ignored, but really actually intrigued me. And actually, we presented this in Journal Club, and people pointed this out, that that in Enterocollitica, what they found is you knock out Invasin, that the bacteria actually have very poor colonization of Peyer's patches, but still cause systemic disease like a wild type does. So therefore, what's going on here is that it sounds like there's actually two infections going on. Invasin's actually mediating entry into the Peyer's patch, but that's not necessarily causing systemic disease. The systemic disease that's going on is going through some other site. And that was actually very clear, actually. And actually, Jeff Pepe dropped out of science and became a lawyer. But this actually turns out to be one of the most important discoveries of that color the way I think about enteric pathogens, actually. And then Joan, while she was still in Stan Falkow's lab, actually showed that Invasin mutants had very poor colonization of the cecum. That hasn't, and that was connected to delaying colonization of the spleen. And I'm not sure that's totally been explained, but I think that when you see colonization of the cecum, that what's happening is that's colonization in which the bugs are growing in Peyer's patches and then just kind of dumping back out into the intestinal lumen. Okay, so so this again, this is Jeff Pepe's result, and what you can see is this is an Invasin mutant. This is a wild type, and what he showed was there's very poor colonization in localized in localized tissue sites. Um, but then if you look in the liver and spleen, what you can see is that you can still see bacteria getting the liver and spleen. A little bit delayed kinetics relative to what you'd see with the wild type, but basically the same. And it takes 4 days before they see bacteria in the Peyer's patches with the N mutant. Okay, so that was all. We could all reproduce that stuff in pseudo TB. So Penn Bargs picked this up, and actually Molly Bergman did this experiment, and what she did was took a mouse mutant that's a lymphotoxin beta knockout mouse, and these guys don't make Peyer's patches, and then asked what happens with systemic disease. And so you can see with the wild type, of course, there's bacteria in the Peyer's patches. There's bacteria in the mesenteric lymph nodes. There's bacteria in the liver and spleen. And here's what this is again with a wild type organism. And here what you can see is that there's no Peyer's patches to look at. In the mesenteric lymph nodes, most of the mice actually don't have any bugs there. And then for most of the animals, not all of them, okay, there's still a few animals that don't have bacteria there. Most of the animals actually do have bacteria in the liver. So basically what this is saying is that there's a root independent of the Peyer's patches, obviously independent of Invasin, that is by and large responsible for systemic disease. We have some other weird things here, like actually we do occasionally see animals, and I'll talk about these in a little bit, that don't colonize in the intestine even. We don't totally have an explanation for that in these lymphotoxin mutants. And if they don't colonize in the intestine, actually you don't see anything going on. That actually led to this model. Actually what's going on is the bacteria need to colonize in the intestine before they cause systemic disease, but they don't need to colonize in the Peyer's patches. All right, so then this comes to the idea, this now is the first clonal analysis in tissues that's ever been done. And people do this now fairly frequently because of deep sequencing, but this is before deep sequencing existed. And so basically the idea here is follow the fate of bacteria from different tissue sites by barcoding the bacteria. And so the idea here is if bacteria are moving from the Peyer's patch into deep tissue sites, if you have a bunch of bacteria that have different barcodes, and I've just colored these different colors, you can determine whether the bacteria from the Peyer's patch go into different tissue sites by whether the barcodes are found in these deep tissue sites. This experiment, we devised this experiment. Penn Barnes actually was a visiting student from Great Britain, and she worked in my lab for a number of years, and she developed this assay. She was an MD. Is really interested in the medical aspects of the infection. And so then the idea was do an infection with a bunch of barcoded bacteria that have different oligonucleotide barcodes, allow infection to go on, then isolate the Peyer's patches, mesenteric lymph nodes, liver and spleen, and then see whether the barcodes in the liver and spleen match those in the Peyer's patch. And if they do, then bacteria from the Peyer's patches have gone to the liver and spleen. And so the way she did this is she would take an input pool of 33 clones, she would infect mice, and then harvest the organs from each one of these tissue sites. And then she'd ask whether the red means they're in only one tissue site and yellow means they're in both or whatever. So that's kind of the way she did things, and then look to see whether bugs that are barcoded from the Peyer's patch are also found in the liver and spleen. All right, and so the first thing she found is that the infection is really clonal, and that is that if you look in the liver and spleen, many of the animals have 1 or 2 or 3 or 4 clones, but not many. So that's really pretty profound if she looks in the deep tissue sites. So basically what that's meaning is that lethal disease is actually being initiated by just a few bacteria. And then if you see the other thing that you see, if you look at the liver versus the Peyer's patch, only 25% of the of the bacteria found in the liver were actually found in the Peyer's patch. But if you look at and compare that to the intestine, 87% of the bacteria in the liver were found in the intestine. So that gave rise to this idea that there's an intestinal reservoir giving rise to the bacteria. All right, so now, so now the idea here is that we don't totally discount the idea that the bugs could be coming from the Peyer's patches. And so then the idea here, and we had evidence for this, because the idea is that there's some bugs coming from the Peyer's patch and some coming through the alternative route. And you know, if we mutationally knock out one or the other, we should still be able to see bacteria getting into deep tissue sites. And we might be able to see this at the single mouse level. So then the idea here is that in an animal where we cut off this flow, what we might see is that in mouse 1, 50% of the bacteria will come through this alternate route. In mouse 2, maybe there'll be no effect because none of them are coming through the alternate route. And then in mouse 3, if we're knocking out this alternate route, there's no flow. So basically, we don't see any bacteria going through that particular route. So this is kind of a soft experiment. We didn't publish this, but this gives you some idea of the way we were thinking. So if you look at the lymphotoxin beta knockout, one of the things you could see is that, you know, in about half the animals, you could see this in the previous experiment, a certain proportion of the animals actually, 1, 2, 3, 4, I guess 4 out of 15 actually, we could see no bacteria in the liver and spleen. So that didn't really, the idea was that there's an alternate route outside of the Peyer's patches. So the expectation was that we'd see nothing down here, but it turned, we would see no evidence for a different route, but we did see some. So there were some bacteria that didn't get to the liver and spleen. So that didn't totally hold to the model. The idea is that there is a small fraction of bacteria that may come through the Peyer's patch, and these guys are being kind of knocked out in these different animals. But then there are these other mice in which going to the liver and spleen is unimpeded. And then there's another group which kind of see lower amounts. And so that's where we kind of think of this as kind of a three-group problem, where we have, in the lymphotoxin knockout, we have a group of mice that simply don't get sick, and they're at low frequency. And of the ones that do get sick, they're of 2 different kinds. Ones that look just like the wild type, and the other that kind of have 50% there, because these are the animals where you get flow from 2 different sites. So again, we didn't publish this concept, but it was kind of the idea of what's going on in the lymphotoxin mutant. All right, so so now this gets to the YOPs, and actually this is kind of an interesting thing. So this is an example of where we went with Yersinia after this, and I got interested in innate immunity in the, you know, about 15 years ago. And as we were working on this, one of the things I became kind of concerned about is that the innate immunity field seemed like it was really married to this concept of PAMPs, which were pathogen-associated molecular patterns. And actually, there was nothing specific about these pathogen-associated molecular patterns. They're actually patterns that are found in all bacteria. And of course, PAMPs are any number of things: LPS, lipoproteins, peptidoglycan, nucleic acids, which, you know, from bacteria, LTA from gram-positive flagellin. And they're toll-like receptors, which are on the surface, and in phagosomes, which are able to recognize it. In addition, one could argue that the cytoplasmic ones do detect pathogens, because in order for them to recognize patterns from a microbe, and NLRs recognize a number of things. The NOD receptor is most famous for recognizing peptidoglycan fragments, and RLRs recognize, Rig-like receptors recognize RNA. One could argue that these guys are really pathogen specific, because the pathogen molecules would never be able to get in there. But most TLRs don't behave that way. TLRs in general are pathogen agnostic. Okay. And so at that point, I sort of started this model and actually presented this first in Israel in 2007. And Dan Portnoy was there, and that gave rise to this pathogen patterns model. And so the idea was that pathogen patterns distinguish pathogens from harmless organisms. And I might say that actually information on this also exists in the plant literature. So people in plant literature were already thinking about things this way, although people who worked on mammalian innate immunity were not. And so then the idea here was that when a bacterium or a pathogen encounters a host cell, a lot of things can happen, and they disrupt translation. They can disrupt translation. They can disrupt the cytoskeleton. They can misregulate channels. They can hijack trafficking and whatnot. And so then the idea is that these themselves, the stress responses that occur as a result of these various disruptions, are themselves patterns that allow the host cell to actually recognize that a pathogen is there. And so Dan went back to Berkeley, and Russell Vance was already thinking about things this way. And so then we wrote this review article. Basically, this paper kind of resulted from this talk that Dan got really excited about. Okay, so this is now the interesting thing about this is this was all based on an idea that the type 3 secretion system was directly causing, was stimulating an innate immune response due to the insertion of the type 3 translocators. And what I'll show you is that recently was shown that wasn't the explanation for what was going on. Actually, there's a very different one that was going on. And actually, what explained our data was that there was a novel PAMP, a novel small molecule that's produced, normally in gram negatives, which gets translocated through the channel and was causing an innate immune response. Okay, so this is work started by Vicki Auerbach. She was actually, had been a graduate student in Dan Portnoy's lab. At one point, I had a very Portnoy-heavy lab. Vicki was in the lab, and Sina Mohammadi, who was a technician in Dan's lab, and then Greg Crimmins. I don't think they overlapped. He overlapped with Vicki, but he quickly came in my lab, and he was also a graduate student in Vicki's lab, from Dan's lab as well. So we had sort of from 2005 through 2013, my lab had very close connections to Dan Portnoy's lab, both intellectually as well as personnel-wise. All right, so again, the type 3 secretion system produces YOPs, and everybody knows that the YOPs terrify the host cell and cause rearrangements. And so what Vicki was interested in doing was identifying response to the type 3 secretion system in the absence of YOPs that occurred in the absence of most known pattern recognition molecules. And the way she did her experiments was the following. So she did the experiments by taking Yersinia, that was the same strain that Kristen uses in most of her experiments, that is missing all the YOPs, and called delta 6. It's called delta 6 or delta 5, depending on how you feel about YOP T. And then this delta 6 strain then isn't making the YOPs, but it does have a translocator. And then what she did is she then infected under conditions which are missing TLR receptors function because it's a mighty 88 knockout, as well as missing most recognition of gram-negative peptidoglycan because it's knocked out for NOD1. So these were double knockouts, macrophages that we got from Golenbach, from Doug Golenbach's lab. And so, and this is an example experiment. So this is what happens if you just look at TNF alpha transcription levels. If you have uninfected, you have the delta 6, you see a robust expression of transcript. And if you don't have the one of the translocators there, so it actually has the type 3 secretion system, but is lacking one of the translocators. Excuse me, I just dropped something. Then what you can see is that there's no response, and that no response looks the same as having no type 3 secretion system. And so, she mapped this in detail. These are arrays that she did. Whoops, there's some. shows you one of the rays she did. So this is what it looks like, comparing the delta 6, which also has a deletion in the translocator, and comparing that to what this looks like in just the absence of the YOPS, but in the presence of the translocator, infecting into strain that's missing TLR function and missing NOD1 function. So there clearly was a innate immune response, which allowed a huge number of genes, and most of these were NF-kappa B regulated at the 2-hour time point, at the 4-hour time point. They were regulated by both NF-kappa B as well as interferon. All right, so that gave rise to a lot of interesting experiments, and so Andreas Baumler and then Russell Vance directly showed that pathogen proteins actually do induce an innate immune response. Actually, this one actually is NOD1 dependent. One of the salmonella effectors, I think it's SOP-B, which activates SOP-E, which activates the SOP-B, which activates Rho-GEFs. Also, that activation is detected by NOD1, and then you get an innate immune response, totally dependent by the translocated protein. This one's a little better. This is actually a more recent paper, and this one actually shows that anthrax toxin directly activates a NOD-like receptor by cleaving it, and then activating it, and then giving you an innate immune response, which is kind of indistinguishable from what you'd see from like LPS or some other molecule. Okay, now was Vicky right, or was I right that this was an innate immune response? So Vicky actually went back to her lab and decided what she would do is she tried to convince people that maybe the translocator itself wasn't being it wasn't, the response you were seeing was not due to translocator, but actually maybe there was a innate immune molecule from the cytoplasm of host cells that was being translocated through the translocator, and she could not get that funded. So she stopped working on it. And then a few years later, Feng Xiao then wrote me, and he asked me for Vicky's strains, the Delta 6 strain, and the one that was Delta 6 YOP-B. So I gave them to him, and so then he then asked the question, what's going on here? So, you know, the initial idea actually was that the translocator was causing the stimulation to cause TNF. The alternate model actually is it's not the translocator, but there's some small molecule being produced by Yersinia that's going through the channel and activating the host innate immune response. And Vicky believed that, but just never got funded and just dropped working on it. Okay. So then you may have seen this paper about a year and a half ago. Feng Xiao then took this strain and then did a transposon mutant hunt, and then just looked for transposon mutants that didn't did not activate NF-kappa B. So he used an NF-kappa B reporter that was luciferase activity, and then screened for mutants that failed to activate. And he found that the transposon mutant that he got that failed to activate is right here, okay, compared to right here. So this is NF-kappa B activation, which is kind of parallels to TNF activation. Was actually in something that was involved in lipopolysaccharide biosynthesis, and it turned out to be bacterial. It turned out to be ADP heptose. And actually, another lab had found that that something similar, that phosphoheptose that was actually an activator of that heptose phosphate was an activator of innate immune response. And that turns out that was probably an error. What they were looking at was some degradation product of ADP heptose, and the phosphoheptose binds alpha kinase 1 with a low affinity and activating the innate immune response. And then ADP heptose is the actual signaling molecule. So we now think that that this result actually, although it may have stimulated some interesting papers, the actual reason for this result was due to ADP heptose actually going through the channel. Okay, so the last thing I just want to talk about here is something that we don't do in the lab much. I just wanted to bring this out, and that's FRET. So I got really intrigued by FRET in the early 2000s, and Cao Wing would just do anything. I mean, he just, um, he was, graduate student, started my lab in 2000, and he had already published a pretty good paper in the Journal of Experimental Medicine in 2003, and was looking around for things to do. And I got really interested in just looking at what the behavior of activation of RAC1 was in response to engagement of integrin receptors. And so we use this basic system, which I actually like better than some of the FRET that we're doing. And these are two protein systems where we have, I'll show you in a second why I like this. So we have one protein linked to CFP, another linked to YFP. And if these two proteins interact, then we would get a FRET signal by exciting CFP and then looking for emission that would then stimulate YFP. And if the two proteins interact, then we get emission, and if not, we wouldn't get emission. And so the way we do our experiments, again, is a little different than people do them now. People like to use single protein fusions, which actually causes problems that we can solve by using two protein fusions. So the way we did this was by taking RAC, which is a protein that controls the cytoskeleton and is activated in response to integrin engagement by Invasin. In the presence of a small binding protein from this protein PAC65, which is a cytoskeletal protein, which will only bind RAC1 when RAC1 is in a GTP-bound state. And so if RAC1 is not activated, then there's no FRET that occurs in the cell. And if RAC1 is activated, you see binding, and you can get spatial information out of this by doing microscopy. So you can see where RAC is activated in the cell. And so we did this with this experiment, and I'll show you a different way that Sina Mohammadi did this, even easier than this. Okay, so this is an example of the way, and so one of the things that Cao Wing found is that we could get activation of RAC. It was spatially regulated, and this actually will show you why I actually like doing things this way. So this is an example of staining for bugs. This is the fluorescence. This is the CFP fluorescence. And this is the YFP fluorescence. And then this is the sensitized FRET. So one of the reasons why I like doing this experiment is you can kind of see what's going on here. So the way this was done is this was, we do experiments where we excite the CFP and then look for emission from the YFP from PBD, okay? So one of the things when you do experiments like that people often don't take into account is that when you excite in this fashion with the 440 nanometer light, it actually excites the YFP a little bit as well. And as a result, you get some emission in the 527 nanometer. So when people do single protein fusions, they're not able to take this into account. So we were able to take this into account by doing controls in which we look for the amount of intrinsic emission from YFP after exciting at 440 in the random cells in which we're only getting single transfection of the YFP PBD. So it's kind of one of the reasons why I like this strategy. So this gives you an example of the sensitized FRET. Again, you can't see FRET. You have to do the subtractions and the calculations to do this. This takes into account the aberrant emission of the YFP signal due to the excitation of the CFP laser, this CFP excitation. Okay. All right, so then you can see that it's restricted bacterial sites. And this gives you an example of these calculations. So this shows you when we normalize this to this other interesting thing is if you normalize it to the amount of RAC that's there, you actually see activations above background, which is kind of interesting. That means that not only is there RAC recruited, but the concentration of RAC that's recruited is activated highly relative to other places in the cell. What we're showing here is this is comparing constitutively active RAC to RAC that's activated when it's around at the bacterial entry site. So Sina actually, searching around the literature, actually found there was an easier way to do things. This is probably one of the reasons why we don't do much for that in the lab anyways. And Joel Swanson had shown that you actually don't have to do that. One way of actually looking for interaction is take this PBD, and it will be recruited to wherever the activated rack, in this case CDC42 is, and then you'll get this fret. But you actually don't need to do fret. Since this interaction between CDC42 and the cell binding domain, if this gets localized, that's enough to say that there's activation. So you can actually follow activation rather than following fret. You can actually follow activation by taking these binding domains, which will only interact with row family members that are activated at the activation site, and just look for recruitment of these cell binding, CBD means CDC42 binding domains, to where you have active row family member. And so this is exactly what he did. So this isn't using fret, but what he showed that in real time you can do this. And this might be the reason why we don't, we kind of stopped using fret, because we could do it in a non-fret fashion, which all we're doing here is simply looking at the localization of a protein that would not be localized around the bug, unless you have an activated row family member in the region. So this just shows you, this is just unfused cherry, and this is then the same thing, where we have CBD actually linked to it, which is the CDC42 binding domain. Okay, so that's all I have to talk about, okay? So that's the whole history of the Yersinia part of the lab.